Hello, everybody. Today I want to talk to you about some novel research that we're doing in the lab. And we have uncovered a new process that we call Ribothrypsis, and it has to do with mRNA decay in cells. And I think this is very relevant to this conference because this is something that we're going to have, all of us, we're going to have to think about analyzing RNA-seq experiments and in general RNA gene expression profiling data. So typically, in any gene expression assay, and particularly RNA-seq, one of the main assumptions that we make is that the RNA that we're getting exists largely as full-length molecules in cells. So we isolate RNA and we do the fragmentation step, and then we sequence by synthesis. We assume that these fragments are coming from full-length molecules. So we make this assumption actually because this is the textbook view of the mRNA lifecycle. So we assume that canonical mRNAs have a 5' cap, they have the 5' UTR coding sequence, 3' UTR, and the poly-A tail, and ribosomes are recruited and translation initiates. And at some point, there is decapping and deadenylation of the mRNA, and then there is mRNA decay. And these two processes are largely considered two distinct processes. Now, of course, we know that for defective mRNAs in cell, there are mRNA surveillance systems that recognize and destroy these defective mRNAs by initiating endonucleolytic cuts. Recently, this distinct role of mRNA decay and translation has started to be questioned by several groups, actually, both yeast and plant. And it has been shown that XRNs actually degrade mRNAs co-translationally following the last translating ribosome. But of course, it is unclear what happens when trimming is initiated at the 3' end of the mRNA, and in general, what is the prevalence and features of co-translational mRNA decay, and even if it happens in humans. So to address that, a critical step is to be able to identify the 5' end and the 3' end of the mRNA simultaneously. So what we did is we developed a new protocol, which we call Acron-seq, and basically consists of two sub-protocols, Acron-3 and Acron-5. And in Acron-3, we sequence the 3' ends of capped RNAs, and on Acron-5, respectively, we sequence the 5' ends of polyadenylated RNAs. So in both cases, we know where the 5' and the 3' end of the mRNA is. Now, if everything was according to what the textbook view of the mRNA is, what we would expect, for example, for the Acron-3, remember these are all capped RNAs, we would expect to see all the 3' ends being at the extreme end of the 3' end. But this is not what we see. Actually, what we see is that there are a lot of ends within the mRNA body, and this is true for the Acron-3 and for the Acron-5 as well. And actually, what we see is that approximately 60% of the captured mRNAs have 3' ends within the coding sequence. So this indicates that only few RNAs actually in cells are full length, and there's very, very extensive in vivo degradation. Now, we wanted to see what is the relationship and if the creation of these native mRNA ends has anything to do with translation. So for that, we use ribosome profiling data, which basically ribosome profiling data is a steady-state view of where the ribosome sits on the mRNAs and where it spends most of the time. So one of the most informative analyses that we did for this process is to compare where we see the Acron-3 ends or where we see the Acron-5 ends with respect to where we see ribosome profiling data, ribosome profiling footprints. And if these two processes are actually not related, then we would expect to see something similar to a randomized experiment where we see no signal around position 0 here, which position 0 corresponds to the 5' end of ribosome profiling data. So, however, when we actually plot the real data, this is what we get, which is completely different from the random experiment and from the randomized view. And there are a lot of things going on in these plots, but perhaps the most striking thing that we see in this plot is that the signal keeps repeating itself, and there is some periodicity in the signal. And if we actually measure this periodicity, and we also use discrete Fourier transformation for that, we see that the period in the signal is exactly 3 nucleotides, which corresponds to the ribosome movement. So what we're seeing here is the process that is probably coupled to the ribosome. So we wonder if this is something that's only coming from only few mRNAs. So we split our data into 10 quantiles the mRNAs into quantiles according to their ACRON 5 and ACRON 3 expression, and we basically see the same thing in all quantiles. And also, we isolated the 50% more expressed transcript, and we see that in 63% of them, we see ACRON 3 ends within the coding sequence. So as I said, there are a lot of things going on in this plot, and we put a lot of thought trying to devise a model that would explain, a mechanism that would explain the peaks that we see in these plots. So in hindsight, the model is quite simple. So we assume that as the ribosome translates the mRNA, at some point we assume that there is a stall. So when this stall happens, the ribosome actually triggers an upstream endonucleolytic cut. And this fragment that is created has a poly-A tail, so it's captured by the ACRON 5 protocol. And the 5' end of this fragment coincides with the 5' end of the ribosome profiling data. So that's why we see this peak at position 0. Of course, the consequence of that is that at some point, the upcoming ribosome is going to reach this cut site. And when that happens, this ribosome is also going to stall. And when this ribosome stalls, it's going to trigger an upstream endonucleolytic cut again. Now, this fragment that is created here doesn't have a 5' cap. It doesn't have a poly-A tail. So it's missed by the ACRON 3 protocol. And this is why we see this depletion here of 3' ends exactly at the cut site, at the cut 1 site. And of course, the consequence is that there will be iterated cuts by upcoming ribosomes until the whole mRNA is degraded. So we term this process Ribothrypsis, which is basically a ribosome-phased mRNA decay during translation. Interestingly, the model makes some testable predictions. The first prediction that it does, and I think it's very interesting, is that if Ribothrypsis is actually a ubiquitous process, and then the Ribothrypsis ends that are created by the process should be present in RNA-seq from total RNA without digestion. And an RNA-seq experiment that fulfills these requirements is a small RNA-seq experiment, where there is no digestion of the RNA and just size selection. So when we actually mine these small RNA sequencing data and we compare them to ribosome profiling data, we see exactly the same profile, and we see this 3-nucleotide periodicity, which indicates that small RNA-seq data actually retain the scars of co-translational Ribothrypsis and exhibit the periodicity with the elongating ribosomes. The next predictions that are actually coming from the model is that what we're seeing should be independent from any 5 to 3 exonuclease, including XRN1. And the reason for that is because the signal that we see in these plots are coming from capped RNA. So there is no way that their process is coming from the 5' end. Similarly, it should be independent from any 3 to 5 exonuclease, including the exosome, because the signal that we see here is coming from polyadenylated RNA. So there's no way that something is coming from the 3' end. Now, I don't have time to go through the data, but we used small RNA-seq data upon knockdown of these elements, and we see exactly that nothing changes. What I wanted to show you, which is also very interesting, is I want to focus on this part here, that the final Ribothrypsis product, which I said that we miss from our protocols. And if these final Ribothrypsis products actually exist, there should be indeed present in small RNA-seq data if we mine the really small RNA-seq, and their ends should be upstream of where we see the cuts. So we did exactly that, and we isolated from small RNA fragments that are smaller than 17 nucleotides. And what we see is that their 5' end is exactly 16 nucleotides from the cut site, and their 3' ends is exactly upstream of the cut site, which validates another prediction of the model. Now, we've seen that everything fits until now, but there is also a very critical missing link here, which is the first assumption of our model, which is the stall. So we tried to mine our data and try to find any evidence for that stall, so we could get a better insight about what happens in the process. So what we notice is that there is a small but noticeable G enrichment immediately downstream of where we see ACRON 5 cuts. And we hypothesized that maybe G quadruplexes, which are stable structures of stacked Dugan tetrads, might play a role in the process. So what we did is we did a genome-wide prediction of G quadruplex structures. And when we compare them with where we see the cuts, what we find is that a very, very big enrichment of G quadruplexes immediately downstream of where we see the cut. And not only that, but RG4s that are located in open reading frame 2 have the strongest enrichment for cut. Now, this was quite shocking, and we tried to look into the literature to find any supporting evidence for that. And what we find is that a very recent report published last year, 2016, which indicates that in an mRNA, if we introduce a G quadruplex on open reading frame 2, this has a very, very strong protein suppression. And in the paper, they couldn't actually explain why this happens, but I think this makes perfect sense with what we're proposing here, because these mRNAs are predicted to be rapidly degraded. So it makes sense that we will have less protein product. Now, of course, this was based on a prediction of RG4s, but we also used data from 2 different labs, Ballas-Brahmanian lab and Bartel lab, with RG4-seq, and we mostly see the same thing. We see enrichment of RG4s immediately downstream of Acron 5 ends. Interestingly, what we also find is that the areas where we have the cuts are conserved. And not only that, but the areas where we also have an RG4 there are even more conserved, which indicates that this process, there might be some regulatory role for this process. Now, we don't know exactly what that regulation is, but I think this is something that we need to investigate more. Finally, we also did the analysis for yeast, and we basically see a similar thing. So we hypothesized that Ribothrypsis also operates in yeast. And while I still have your attention, I would like to thank the people that contribute in this work, particularly Zyssimos Mourelatos and Fadia. She did tremendous amount of job for the Acron-seq protocol, designing the protocol and performing it, and of course the funding agency. And I would like to close with a slide that I like a lot, which is that sequencing data are actually snapshots of biological processes. And I think the synthesis and integration of these data is the key for the discovery of biological processes. And I think that if we actually synthesize data appropriately, we can see biological phenomena as if we were seeing a movie. So I will close with the last slide with the key takeaway points. And the first one is that Ribothrypsis mediates ribosome-phased endonucleolytic mRNA degradation of translated mRNAs. That RG4s likely trigger Ribothrypsis. And most importantly, that mRNA fragments persist in cells. And this has a big implication for the interpretation of RNA experiments, including RNA-seq and clip-seq, where we have to maybe redefine what we are actually measuring with these protocols. And with that, I would like to thank you for your attention. Any questions? Thank you.